about two experiments that were done by Robert Emerson and his colleagues, one of which was the flashing light experiment, where two rather unusual conclusions were reached. The first of all, remember we're plotting flash intensity, number of photons that were absorbed by the sample versus oxygen produced per flash. And so if we look at the initial slope of this line, the slope of that line is oxygen per photon absorbed, right? And it told us that the production of one molecule of oxygen required the absorption of 9 to 10 photons. And this was totally unexpected. They sort of thought there was one chlorophyll, one oxygen, one photon. And we'll see where this becomes important in just a second. The other thing they noticed was that if they looked at the maximum flash yield of oxygen production and just took the maximum amount of oxygen produced and divided by the amount of chlorophyll in the sample, they found that on the order of 2,500 chlorophylls cooperate in some way. That the maximum yield of oxygen evolution suggests that a large number of chlorophylls cooperates in some way in oxygen evolution. So as we go on to the next points that we're going to talk about, one of the things we want to do is make sure that we can understand where these numbers come in our larger picture of photosynthesis. The other experiment we discussed was the enhancement experiment, also done by Emerson, where he looked at the amount of photosynthesis that occurred in limiting far red light, greater than 680 nanometers, and the amount of photosynthesis that was done in limiting red light, less than 680 nanometers, and then added them together. And the expectation is that if you get this much photosynthesis from one and this from the other, you add them together, they should give you about this much. He saw more than twice as much. There was an enhancement when red and far red light were given together, much more than was expected from the sum of the two individual ones. So this enhancement effect is something else that we also need to take into account. All right, so now we're going to leap forward about 40 years from the experiments that Emerson did to our understanding of photosynthesis, where it was about the time that I got interested in the mid to late 60s. So this is the biggest, most important picture of photosynthesis that you should be thinking about from the perspective of energetics. So how do we get energy on this diagram? The y-axis represents the midpoint potentials, the redox midpoint potentials of the various components of the electron transport chain. And don't worry, the one thing that you don't have to know in this picture is all this alphabet soup. You know, all these different things. You don't need to know the gory details of those. Too hard to see that? Is that better? Okay. So what do we mean by the energy on this diagram? At the top of the diagram, things with very negative midpoint potentials are good electron donors. These are things that like to give up electrons. And at the bottom of this diagram, we have good electron acceptors, things that like to accept electrons. So not surprisingly, we should expect that the direction of spontaneous electron flow is from good donors to good acceptors. So even if you ignore all the sort of equations of redox potentials and stuff like that, you don't need to know that. You just need to know that things with negative midpoint potentials like to give up electrons, things with positive like to accept electrons. And that the way we draw these diagrams, spontaneous electron flow is downhill. Right? Okay. So if we think back to the overall equation for photosynthesis, for the light reactions that we talked about, we said that we're taking 2 molecules of water and converting that to 1 molecule of oxygen plus 4 electrons plus 4 protons. And then we're using those 4 electrons and 4 protons to reduce two of these. So water is being oxidized, losing electrons to produce molecular oxygen. And those electrons are being passed to this electron shuttle, NADP+, reducing it to NADPH. So in the scheme of these reactions, here's water being oxidized over here. And here's NADP being reduced to NADPH over here. And the first thing you should notice is that it's energetically uphill to go from water to NADP. Right? No surprise there. We know that photosynthesis requires an input of energy to make this work. And so one of the things that we need to understand is how does this energy transduction occur? And it occurs because in photosynthetic electron transport from water to NADP+, there are two inputs of energy. These upward arrows here represent light energy that is input into the system so that once you form good electron donors, good electron acceptors, good electron donors, good electron acceptors, all the rest of electron transport is downhill. It happens spontaneously. So let's look at these two different light-requiring reactions and see what's going on. So in photosystem 2, one of the things we should designate is the idea of a reaction center. Reaction center is just a chlorophyll molecule. But it's a very special chlorophyll molecule in that it is the chlorophyll that takes the energy of the excited state that was formed by photon absorption and converts that energy into oxidants and reductants that drive electron transport. Okay, so the reaction center is sort of the business end of photosynthesis. And for photosystem 2, the reaction center, we abbreviate it RC, is called P680. That's because its absorption, peak of absorption in the red is at 680 nanometers. And for photosystem 1, the reaction center pigment is called P700 because its absorption is at longer wavelength at 700 nanometers. So right away you should start to see information that's relevant to the enhancement experiment that Emerson did. And we'll come back to challenge you a little bit about that in just a minute. Okay, so let's look at what's happening with light energy in photosystem 2. Light energy in photosystem 2 creates an electron donor and an electron acceptor. In photosystem 1, the same thing happens. Absorption of light creates an electron donor and electron acceptor. And all the rest of electron transport is spontaneously downhill. Let's look in photosystem 2. It's downhill from water to the electron acceptor of photosystem 2. So once this light energy has done its thing, oxygen can be produced by removing electrons from water. In photosystem 1, it produces a reductant, an electron donor, that is a good enough electron donor to reduce NADP to NADPH. So two of the main products of this are sufficient to oxidize water and to reduce NADP. And the rest of electron transport is just taking the electrons that go up here and transporting them down here to replace the electrons that were removed in this direction. Okay, so the way we want to think about photosynthetic electron transport is that it is two inputs of light energy that function in series. That's the important part. They function in series. So that both photosystem 2 and photosystem 1 need to absorb light energy in order for this process to work. So let's go back now and think first about the enhancement experiment, which said if we illuminate with light that's at wavelengths greater than 680, this should be hitting photosystem 1, this should be greater than 680. This is hitting photosystem 1 primarily. If we use light that's less than 680, This is hitting photosystem 2 primarily. And if we put them together, we get more light and more oxygen evolution than you'd expect. How do we explain that? How does this picture of photosynthesis explain the enhancement effect? Right, so you can't run these reactions by themselves for very long. So for example, if we only excited photosystem 2, then there's no place for all these electrons to go. You can take electrons away from water only as long as there's some place for all those electrons to go. If you're not hitting photosystem 1, it runs out of gas pretty quickly. The same is true of photosystem 1. You can reduce NADP, but you got to replace those electrons that are lost from somewhere. So if you're hitting one or the other of these individually, you see a little bit of oxygen evolution, but not very much. But if you hit them both simultaneously, then you see lots more oxygen evolution than you'd expect. Okay, let's go back to this experiment. In particular, let's think about this quantum requirement of 9 to 10 photons per oxygen evolved. How does that come out of this diagram? This is a little more complicated. Emmy? Well, so it takes 8 or 9 or 10 photons right here.
Well, I think also that's 702.
Okay, let me simplify the question a little bit. What would be the minimum number of photons based on this mechanism that would be required to produce one molecule of oxygen?
8.
Why do you say 8?
Because I need one to transport the first electron up to P60, 680 excitement, and then another one to get it to P70. That makes 2, and we have 4 electrons.
4 electrons, right. Yeah, so this tells you that the minimum quantum requirement should be 8, right? 4 here and 4 here for the 4 electrons that are moving along this whole chain. So 9 to 10 is what you'd expect sort of from a thermodynamic perspective. There's no such thing as perfect energy matches. Some of that energy has to be given up as heat or some way to increase entropy, right? So that quantum requirement of 9 to 10 is due to the fact that this process isn't 100% efficient. Some of the energy's got to be lost in the environment to increase the entropy. Okay? So this is the sort of thing that I want you to be able to do with the information that you've been given from your readings and that we talk about in lecture, is see how things fit together in a logical way. Don't treat them as independent things. But certainly knowing all the alphabet soup here, I mean, if you take my photosynthesis class, we talk about all these things in great detail. But it's not necessary that you know those. I'm not going to ask you a question, what comes after plastoquinone in the electron transport chain, because it's just not important that you know the names of these things. But it is important that you recognize that photosynthesis is basically, you can describe the light reactions as 2 inputs of light energy, photochemical reactions, that act in series to provide the energy to drive electron transport from water uphill to NADP. So it's storing energy in these compounds, NADPH, that we know is going to be used for CO2 fixation. The only thing that we're missing in this whole picture is ATP. Because remember, we said per CO2 that's fixed in the dark reactions, we need 2 NADPH and 3 ATP. Where's the ATP coming from in this diagram? There's no ATP in here. But what we'll see is that this electron transport not only provides the redox energy to produce NADPH, but also socks away some energy that can be used for ATP production. And we'll see how that happens in just a minute. Okay, so the key thing to understanding this is to recognize that these light-requiring reactions produce oxidants and reductants that make all the rest of electron transport happen spontaneously. And if you're interested in the gory details of that, come see me in my office hours and I'll make you crazy thinking about all that stuff. Okay, I want to spend just a minute talking about the part of photosynthesis that I spent basically most of my life working on, and that's the antenna function of photosynthesis. And if we look at this diagram from your textbook, it's really sort of meant to give you at least a qualitative idea of what's going on in the antenna. Let's think about it this way. Let's think about light as being raindrops. I mean, photons. Think of photons as being raindrops falling down. And this is P680 or P700. It's a reaction center that's going to do the business of photosynthesis, driving the reactions that we just talked about. If it's raining lightly, then relatively few drops will hit this reaction center in any given amount of time. And that the number of drops, the number of photons that hit this reaction center will determine how fast photosynthesis can happen. In the brightest sunlight, so the highest light intensities that are naturally encountered on the surface of the Earth, the rate at which a single chlorophyll absorbs photons is on the order of 5 per second. Sounds like a lot, but the capacity for electron transport through here is about 1,000 per second. So that means that the highest rate of light absorption by a chlorophyll is grossly lower than the capacity for electron transport. How do plants and photosynthetic organisms get around that? Instead of having just one pigment out there absorbing energy, they take hundreds of these guys and lay them out there. So if this is one, if we have 100 of these, then the rate of light absorption by the antenna is going to be 100 times greater. And the only requirement of the antenna is that the energy that's created in the excited state is somehow transferred to the reaction center where it can be used in photosynthesis. And so this is the part of photosynthesis that I study. What's the mechanism that allows this transfer to happen? Because this transfer is very efficient. There's very little energy loss in this process. All the energy losses in photosynthesis happen in these electron transport steps, not in these energy transfer steps. The time scale of excited state moving from one chlorophyll to another is on the order of 10 to the minus 14th seconds. The time it takes the energy to move through the whole antenna and get to the reaction center is on the order of 10 to the minus 10th seconds. So it tells you that the energy is moving around a lot in the antenna before it gets to the reaction center. Thousands and thousands of individual hops through the antenna before it's used in the reaction center. Okay. So this is nothing more than the name suggests, an antenna. It's increasing the effective surface area through which light energy can be absorbed to bring the rate of electron transport, light-dependent electron transport, to be commensurate with the capacity of photosynthesis, around 1,000 per second. Okay, so there's a lifetime's worth of work in 3 minutes. Let's go on to talk about characteristics of the chloroplast itself in preparation for moving from thinking about the light reactions, which we'll finish up over the next 15 or 20 minutes, to talking about the carbon reactions. So all of this is going on inside these chloroplasts. And remember, we said that chloroplasts are of endosymbiotic origin. That means that some free living organism was taken up by another one and became a functional part of it. One of the hallmarks of endosymbiosis are these double membranes. The outer membrane of the chloroplast was originally derived from the outer membrane of the host. The inner membrane of the chloroplast was originally derived from the membrane of the plasma membrane of the endosymbiont. Okay, so that these double membranes that you see in chloroplasts and mitochondria are the remnants of endosymbiotic events. These membranes basically separate the cytoplasm from the volume enclosed inside the inner membrane called the stroma. So everything that's happening in photosynthesis is happening in the stroma or in these additional set of membranes inside the chloroplast called thylakoids. Thylakoid in Greek means flattened sac. And you can see that if you look at the structure of these thylakoid membranes here blown up over here, they're membranes that are very convoluted but look like, sometimes they look like stacks of pennies almost. These are called grana stacks. One really interesting characteristic of these thylakoid membranes that doesn't say anything about in the book but is very intriguing. You can very easily isolate chloroplasts and rupture the chloroplasts and remove the envelope and just have pure thylakoid membranes. If you take these thylakoid membranes and you put them in 5 millimolar magnesium chloride, don't worry about the actual components of the solution that we're putting these in. If you put them in 5 millimolar magnesium chloride, they look just like they look inside chloroplasts. You put them in 1 millimolar magnesium chloride, and this very convoluted thing unfolds to form one big spherical vesicle. You put it back into 5 millimolar magnesium chloride, and these stacks and all this complex structure reforms. And this can go back and forth many times over the time scale of, it takes 10 or 15 minutes for that to be complete. Pretty bizarre. And I think one of the important things to ask is, why do these membranes fold up like this? What's the purpose of that? And the honest answer is, we don't know. That was actually what I did my PhD project on. I compared photosynthesis in a certain type of algae where the thylakoid membranes stack up like this, and to other types of algae where they're just single sheets of membrane without all this complex structure. And looked at all the different characteristics of photosynthesis, and they were basically indistinguishable. So what this is actually doing, we don't know. Yes?
So no one is better than the other?
Well, let's put it this way. As far as the questions that we've asked so far, one is not better than the other. The fact that this happens tells you that there's got to be some benefit to this. Otherwise, it wouldn't have been selected for in so many photosynthetic organisms. It's not present in all of them, but it's present in a lot.
So which are the unicellular organisms that have unstacked?
That have unstacked? All the green algae.
But maybe just because the chloroplast, we can understand that, came from the green algae.
It appears that eukaryotic chloroplasts they're not monophyletic, but they're pretty close to monophyletic. So that means it didn't happen a bunch of thousands of different times independently. There was just a few events. Could that be part of the difference? Absolutely. It could be that there may be some evolutionary heritage to this, but you still have to ask the question, why do some organisms have just one sort of flattened out membrane and some organisms have all these really complex stacks? Yeah, but as I said, the comparisons that we made showed there was no benefit to that. Believe me, we tried hard to find some benefits. Okay, so let's just take a second to ask, what's the consequence of this as far as photosynthesis is concerned? And if we look at the organization of these membranes and ask about, for example, where are photosystem 2 and photosystem 1 located in these membranes? The real distinction that we want to make, let me go back to the previous slide for just a second. We take this giant convoluted thylakoid membrane, and we notice that some parts of the membrane are exposed to the stroma, to the volume enclosed in the chloroplast. And other parts are stuck between where two membranes are pressed together. So we make the distinction between stroma-exposed surfaces and oppressed surfaces. And if we look at that in cross-section, interestingly, we see that virtually all of the photosystem 2 is in these regions where two membranes are sitting on top of each other, in between the stacks of pennies, the grana. All the photosystem 1 and all the ATP synthase complexes are found out in the membrane that's exposed to the stroma. So there is a functional distinction. Remember I talked about adding and subtracting the magnesium and it changes its shape? If you take away magnesium, all these complexes randomize in this big spherical piece of membrane. Put the magnesium back. As the membrane stacks reform, this heterogeneity also forms. Again, this is something we know is there, but we don't know what it actually does in terms of benefit for photosynthesis. Okay. The important part of the organization of the photosynthetic membrane is shown in this diagram. So what your challenge is to understand the relationship between this diagram, which shows the organization of the electron transport complexes in the membrane, with this diagram that shows the energetics of how electrons move from one component to the next. Okay? These show two different things. This is a graph that's plotting electron transport from oxygen or from water to NADP as a function of energy. This one is showing the organization of those electron carriers in the thylakoid membrane. Because remember, the one thing that we haven't figured out yet is where the ATP is coming from. So the electron transport is carried out by three main complexes, protein complexes in the thylakoid membrane. Photosystem 2, photosystem 1, and a thing called the cytochrome BF complex. This is actually a complex that's virtually identical to a complex that functions in mitochondria in respiratory electron transport. Very similar structure and function. But just fitting the sequence of electron carriers into this, water is oxidized on the inside. The inside of the thylakoid membranes is referred to as the lumen. It's the volume enclosed by the thylakoid membrane. Water is oxidized to molecular oxygen on the luminal side of photosystem 2. At the other end of the electron transport chain, NADP is reduced to NADPH on the stromal side, and everything else that's happening is, electron transport through these various carriers there. Okay, so we can pretty easily put that sequence of electron carriers into this diagram to get the electrons from water to NADP. The other thing that we have to get out of this diagram is where does the ATP come from? And as most of you know, the key to ATP synthesis in both mitochondria and chloroplasts comes from a guy named Peter Mitchell and his chemiosmotic hypothesis. Basically, what the chemiosmotic hypothesis says is the energy that's needed to make ATP comes from a proton gradient. So it's a proton gradient driven ATP synthesis. We'll talk in just a minute how this was actually proven. When Peter Mitchell first proposed that ATP synthesis was driven by a proton gradient, people thought he was nuts. But he was proven actually by a professor here in plant biology, a guy who's still up on the second floor, is the guy that proved that Peter Mitchell was correct. But let's first of all ask, how is a proton gradient created by this pattern of electron transport? And that's relatively straightforward. We know that the reaction that oxidizes water to molecular oxygen produces protons as a byproduct. So for every oxygen molecule produced, there are 4 protons that are released into into the lumen of the thylakoid membrane. For every NADP that's reduced to NADPH, protons are taken up on the stromal side of the membrane. So simply the oxidation and reduction reactions that are happening at the ends of electron transport contribute to making a proton gradient, releasing protons on the inside, taking them up on the outside. But in addition to that, there's another component to forming the proton gradient, and that has to do with a molecule called plastoquinone, PQ. PQ is an electron and proton shuttle. PQ picks up electrons from the electron donor side of photosystem 2 on the stromal side of the membrane, and at the same time picks up protons. So the PQ gets reduced to PQH2. So it's carrying 2 electrons and 2 protons from the stromal side of the membrane over here to the cytochrome complex, where when it's oxidized, it gives up its 2 electrons but releases its protons on the inside. So the fact that it's reduced on one side of the membrane and oxidized on the other side of the membrane facilitates proton transfer. So basically what we have here is a proton, we have an electron transport that's coupled to proton transport. It's obligately coupled. A part of the electron transport, an obligate part of the electron transport is creating this proton gradient. And the energy of that proton gradient, we know there's energy in it. The energy of that proton gradient can be used to synthesize ATP. This is essentially the opposite process to the proton pump that we talked about in the plasma membrane, right? The plasma membrane proton pump takes ATP, hydrolyzes that ATP, and pumps protons out of the cell. This is the opposite reaction. It's taking a proton gradient, allowing those protons to move down their electrochemical gradient, and using that energy to produce ATP. And surprisingly, perhaps, this whole complex that's involved in ATP synthesis has now been crystallized. We know the structure of it, and we know the gory details, pretty much, of how proton movement through this complex can be used to generate ATP. And presumably that gives us some idea about how the proton pumps work as well, because it's probably going pretty much in the opposite direction. Okay, so the key aspect of this is that the ATP is produced because of the coupling of electron transport to proton transport across the membrane. It creates a proton gradient that then can be used to synthesize ATP. Sure it does. Where's the energy coming from? No. That would be somewhat foolish, wouldn't it? Yeah. Where's the energy coming from? Where's the energy to create this proton gradient coming from? You seem somewhat dubious about that.
I mean, I think it's like, it could be the electron.
Which one?
So remember that part of the picture is that the proton transport is obligately coupled to the electron transport. Where's the energy to drive the electron transport coming from? Where's the energy to create the proton gradient coming from? The same light energy, right? If it's driving the electron transport, it's driving the proton transport simultaneously because they're coupled together. So now we've got the energy converted from sunlight into energy that's stored in NADPH and energy that's stored in ATP. All right, I just want to finish up by giving a brief description of an experiment, a very clever experiment that was done by Andre Jagendorf when he was a student at Johns Hopkins University. He was an undergraduate here at Cornell, and then he came back as a faculty member, but he did his PhD at Johns Hopkins. And he was intrigued by Peter Mitchell's chemiosmotic hypothesis. And so he said, we can prove this really easily. People knew that we could isolate thylakoid membranes. So if we take, I'm going to draw a thylakoid membrane in a very simplified view. That's a thylakoid membrane. And here is the ATP synthesis complex. So there's a part that crosses the membrane, and there's a catalytic part that makes the ATP. So this would be equivalent to the stroma, and this would be equivalent to the lumen, the volume enclosed by the thylakoids. Okay, so if a pH gradient drives ATP synthesis, then what Jagendorf suggested was, let's make an artificial pH gradient across this thylakoid membrane. Let's make it pH 4 inside and pH 8 outside, and then we'll add ADP and phosphate, and if this works, should be able to make ATP. So the only tricky part is, how do you make it pH 8 on the outside and pH 4 on the inside? And he did a lot of playing around with this, but what he found was he could take isolated thylakoid membranes and put them in a buffered solution at pH 4 and leave them there overnight. And protons, you think of membranes not being very leaky to protons. They're not on the time scale of the reactions that we're thinking about. But if you leave them there long enough, it will equilibrate to be pH 4 on both sides. Okay? So essentially at this point, after this equilibration process, it's pH 4 on the inside and pH 4 on the outside. Okay? No rocket science what he's going to do next. He's going to add something to make pH 8 on the outside. So he's artificially created a proton gradient across the thylakoid membrane, and if he adds ATP in the dark, he can make ATP. ADP in the dark, he makes ATP. So by generating this artificial proton gradient across the membrane, he could show that independent of the light reactions completely in the dark, he can make ATP from the proton gradient. And the amount of ATP was proportional to the size of the proton gradient. Okay, so it worked pretty well. Relatively, this is the sort of experiment that we want our graduate students to do. Something that's simple and elegant, that really proves something nice. Obviously, most experiments don't happen like this, but this is a pretty cool one. Okay, so let me remind you, the thing that I really want you to be able to do is understand how this diagram, which shows electron transport in the plane of the thylakoid membrane, proton transport in the plane of the thylakoid membrane, and relating that back to this diagram that shows the energetics of this. Francisca. It still has, the thylakoid membrane is still an enclosed vesicle. It's just one big flat one rather than all this convoluted stuff. So there's still an inside and outside. Okay? Yeah, so a key part of this is to recognize that this membrane, the thylakoid membrane, independent of what type of organism it is, forms an enclosed vesicle. It may be a single round vesicle, it could be one big flat vesicle, or it could be all the folded up things that we see in higher plant chloroplasts. But there's an inside and an outside, and essentially what we have here is electron transport that's going from the inside, where water is oxidized, to the outside, where NADP is reduced, and proton transport that's going from the outside to the inside. So there's vectorial electron transport. How this happens is completely dependent upon how these complexes are in the membrane. Put the complexes in backwards, flip them over, everything would be screwed up, right? So it tells you something about cell biology, that how these complexes are built and inserted in the membrane is very important in maintaining their function. You can't put them in any arbitrary way. They have to be in the right way to make all of this work. Okay? Good. All right, now we're going to switch gears to start talking about the dark reactions, the carbon reactions. All right, so what we need to remember is what's going on, the overall reaction. Taking CO2 plus 2 NADPH plus 3 ATP. And what we're producing from this is C3H6O3, and plus some waters and ADPs and stuff like that. This 3-carbon sugar is called a triose. And we'll see that the main product of the dark reactions is a phosphorylated version of this called triose phosphate. So our goal now for this part of the course is to understand how these products of the light reactions are used to reduce CO2 to sugars, which form the basis of everything else that the plant does. All the organic molecules that are present in a plant ultimately come from carbon that was fixed by this reaction. Actually, all the organic carbon that's present in all living organisms ultimately came from this reaction. Even if it wasn't a photosynthetic organism, I mean, if it was a chemoautotroph, still uses the same pathway that we're going to describe here. So it's pretty common. All right, so the other thing that we need to think about is, so we have the light reactions and the carbon reactions, and together these produce this molecule triose phosphate. That's the product, the 3-carbon, phosphorylated 3-carbon product of photosynthesis. What happens to this triose phosphate? I want to put this in perspective before we think about all the details of what's going on here. So one of the things that can happen in the chloroplast this can be converted into starch. But chloroplasts don't accumulate starch forever. In fact, it's a reversible process. And so any of the starch that's created is ultimately, at some point, converted back into triose phosphate. And this is happening in the chloroplast. The other thing that can happen with the triose phosphate is it can be exported from the chloroplast Now we have triose phosphate out here in the cytoplasm. And what can that triose phosphate be used for? Well, one of the main ones is for sucrose synthesis. Sucrose is the main sugar that's translocated in plants from the photosynthetic tissues to everything else. And the other thing that triose phosphate can be used for is respiration. So remember, we said that none of the energy in the form of ATP and NADPH in the chloroplast gets directly out into the cytoplasm. But as we'll see when we talk about respiration, you can take this triose phosphate, run it into respiration, and generate ATP and NADPH that's needed in the cytoplasm. Or you can transport it as sucrose just down to the roots, and then the roots, sucrose gets used in respiration. Okay, so one of the things that we need to think about overall as we think about what the dark reactions, the carbon reactions of photosynthesis are doing, is this partitioning of the main product of the dark reactions among starch synthesis, export from the chloroplast, converting it into sucrose, or using it in respiration. Right? All of these things represent sinks for the products of photosynthesis. And we need to understand how these sinks are coupled together. Okay, so let's first start off by looking at this part of the picture. This part of the picture right here. Converting CO2 into triose phosphate. How did this reaction happen? The original work on this was done by, well, not surprisingly, let's skip that for a second, was done by a guy by the name of Kelvin. Calvin cycle is named after Melvin Calvin, who discovered it. And the way he discovered it was another relatively clever, simple experiment. He took unicellular green algae that were photosynthesizing in the light, and then quickly took those cells and dumped them into a very acidic solution. Killed the cells, stopped all the reactions, and extracted all the products of photosynthesis. But he used something very, very new at the time. He used radioactively labeled carbon. C14 was a relatively, first became available just before World War II. And what Calvin did was took the cells, illuminated them for various lengths of time, and then put them into the acid and extracted everything out of there, and went to shorter and shorter time periods in order to try to identify what compound did the radioactive carbon show up in first. And the compound it showed up in first is this compound called PGA, phosphoglyceric acid. You don't need to know the names. You don't need to know what the abbreviations stand for. You just need to think about where they fit into the overall scheme of things. If you took radioactively labeled CO2, the first compound that carbon shows up in is phosphoglyceric acid. So that tells you this must be, the phosphoglyceric acid must be the product of the reaction that takes CO2 and does something with it and puts it into organic carbon. Okay? So, in the simplest sense, what we have here, CO2 plus something, we don't know what that something is, goes to form phosphoglyceric acid. CO2 has got one carbon in it. Phosphoglyceric acid has got 3 carbons in it. So how many carbons in X? Pretty easy, 2, right? Well, wait, don't give it all away. So Calvin and his colleagues spent almost 10 years looking for a 2-carbon compound that acted as the electron or the carbon acceptor to make phosphoglyceric acid. They never found it. The reason they never found it is because it doesn't exist. The actual reaction that happens is CO2 plus a 5-carbon compound. That 5-carbon compound, its abbreviation is RUBP, ribulose bisphosphate. And this makes 2 3-carbon PGA. So it was some good biochemistry that helped them figure out what this reaction is. So the acceptor of the carbon from CO2 to make PGA is a 5-carbon sugar. Ribulose is a sugar. Okay, so one of the things that we need to think about in this reaction, we could just take all this PGA and take it off and use it in the cell. Why would that be bad? Why wouldn't that work? Why don't we just run this reaction like this and take all the carbon that's fixed and use it in those various processes that we talked about? Where's this going to come from? This is carbon compound in the cell. If you use up all of this, what's going to happen to the rate of CO2 fixation? Stops, right? So there has to be some way to take some of this carbon and recycle it back to regenerate the 5-carbon acceptor molecule and other of this carbon to export to use for other processes. Right? So there has to be sort of a branch point in this sequence of reactions where some of the carbon goes off to other things in the plant, making sucrose, respiration, whatever. But some of it goes back to regenerate the 5-carbon acceptor molecule. Well, you've got the Calvin cycle right there. That's basically the picture of the Calvin cycle. We'll look at it in a little bit more detail, but that's essentially it. Okay, so here's the figure from your textbook. And the good thing about this figure is it's simple. It doesn't show all the detailed reactions. I don't expect you to know all the detailed reactions. And it also puts its emphasis on the three main phases of this overall cycle. The first phase is carboxylation. That's adding the CO2 in. That's this reaction right here. But remember, another part of what happens in the carbon reactions, in the dark reactions, is ATP and NADPH from the light reactions somehow are used up. And they're used to reduce PGA to triose phosphate. Because remember, CO2 has got to be reduced in here somewhere. Electrons have to be added in somewhere. And that reduction happens in this phase, converting the PGA, that's the product of the carboxylation reaction, to triose phosphates. Uses up most of the ATP and NADPH. Then this triose phosphate can just go off and be used for other things. Some of it can. But the rest of the cycle has to convert some of this carbon in the triose phosphate back to that 5-carbon acceptor molecule that was used to initially accept the CO2. So we're going to talk in just a little bit more detail about these reactions. Not a lot more, because you don't need to know all the gory details to really see what's going on here. So you have carboxylation, adding the CO2, the inorganic carbon into make organic carbon. The reduction phase, reducing PGA to triose phosphate. And then the regeneration phase, taking these 3-carbon molecules and regenerating the 5-carbon sugars that we started off with as the carbon acceptor. Okay, so the figure from your textbook shows the structures. I don't care that you know the structures. RUBP, CO2, makes PGA. PGA is reduced with ATP and NADPH to make triose phosphate. That's the level that you need to understand it. You don't need to know any of those structures. But the ATP and the NADPH are being used, most of it, not all of it, but most of it's being used in these steps right here. Another important thing, the perspective that these diagrams that I'm going to show you now, the reaction mechanisms, are drawn from is what we want to do is take one triose phosphate out of this cycle and use it for somewhere else. How many CO2s need to go in to produce one triose phosphate? 3, right? So ultimately, if we don't look at all the detailed reactions, we just look at what's going in and what's going out. To make one 3-carbon sugar that we export to some other use, we need to put in 3 CO2s. So all these reactions that I'm showing you are from the perspective of fixing 3 CO2 molecules. So we need 3 CO2s, 3 RUBPs. It makes 6 PGAs, and then you need 6 ATP and 6 NADPH to produce our 6 triose phosphates. Okay? Now here's the one that'll make you sick. This is the reactions of the regeneration stage. And it looks pretty messy. And it is pretty messy. As far as I'm concerned, this is a really stupid way to look at this. I have a simpler way. This is one I made up 20 years ago, and I still use it. Here's that same reaction scheme shown in a much simpler way. We're starting off up here with 3 carbon dioxides and 3 ribulose 1,5 bisphosphates. And we're making 6 phosphoglyceric acids. Here's our ATP and NADPH being used to make 6 molecules of triose phosphate. The G3P, glyceraldehyde 3-phosphate, is the same thing as triose phosphate. So here's our 6 products. What we're going to do is take one of these triose phosphates and export it for all the other processes. And we're going to take 5 3-carbon sugar molecules and mix up the carbons and regenerate 3 5-carbon sugar molecules. Same total number of carbons. 5 3-carbon sugars, 3 5-carbon sugars. And I don't need to go into the details, but you can accept that switching carbons among sugars is a really easy reaction to do. No, the free energy change is very small. They don't have huge activation energies. It's an easy reaction to do biosynthetically. So I'm just going to walk through the process, not because I want you to see all the details, because I want you to get a feel for what this switching of carbons between sugars is doing. So let's start with this step right here. We're taking 2 3-carbon sugars and making 1 6-carbon sugar. Just added them end to end, okay? No switching around there, just adding them end to end. And it turns out that this process right here is also an important process in the synthesis of starch. So we'll talk more about this in the lecture on Thursday. So we've taken 2 of these 3-carbon and made 1 6-carbon sugar. So the next thing we're going to do is take this 6-carbon sugar and add a 3-carbon sugar to it. 6 plus 3 is 9. Doesn't make a 9-carbon sugar. It makes 1 5-carbon sugar and 1 4-carbon sugar. So you took 6 plus 3 and made 5 plus 4. Okay? Now we've got 1 5-carbon sugar back, and we've got 1 4-carbon and a couple of 3-carbons left. So we take the 4-carbon and add in another triose phosphate. 4 plus 3 equals 7. Makes a 7-carbon sugar. And then you take the 7-carbon sugar and the last triose phosphate, 7 plus 3 equals 10, and you make 2 other 5-carbon sugars. And then all you have to do is convert all those 5-carbon sugars back to ribulose phosphate. And the last step over here is to use the last ATPs to convert these 3 ribulose 5-phosphates to 3 ribulose 1,5 bisphosphates. So the gory details of this are not critical for you to understand, other than it is just moving carbons around. Just converting 3 5-carbon sugars, 5 3-carbon sugars into 3 5-carbon sugars. Yes?
You have the last GPP marked as the export.
Yep.
And then there's also an arrow. There's also an arrow under the 4th and 5th GCP for another export or storage.
Yeah.
So
Okay, but remember what I said about starch? Starch doesn't stay there forever. It's sort of temporary storage, and everything that goes this way, sometime usually within 24 hours, goes back this way. It goes back into these very reactions, and then other things happen. We'll talk about that more on Thursday. Okay? So the easiest way to think about this, at least from my perspective and from my experience with students, is to view this process as the net reaction of producing 1 glyceraldehyde 3-phosphate, 1 triose phosphate to be exported from 3 CO2s coming in. If you can visualize that, then that's what I'm looking for. And we're looking at a carboxylation reaction, the first stage, the reduction reactions to produce the triose phosphate, and the rest of this is all regenerating the 5-carbon sugar that is the acceptor for CO2. What would happen, looking at this picture now, if all the sugar or most of the sugar that was produced from photosynthesis was exported?
We would run out of the
Yeah, you'd run out of RUBP, and if you run out of that, then CO2 fixation stops. Right? So it's absolutely essential that 5/6 of the carbon that's produced by the reduction stage goes back around to reproduce the RUBP. Okay. The key steps in this, think of this as a giant reaction mechanism, and let's think about it from the picture of energetics. We said that down here, most of these reactions have very small delta Gs. That's right, they're sort of near equilibrium. So how do we make this reaction go in a specific direction? This pathway go in a specific direction? Yeah, so if you use up the products, if we use up this product over here, that's going to help by removing products and driving the reaction in a specific direction. The other way it happens is because some of these reactions have large free energy changes. So the carboxylation reaction, this has a large negative delta G. It only goes in this direction. These reduction reactions have large negative delta Gs. They only go in this reaction. And then these two reactions down here that remove a phosphate from the sugars. There's one here, and there's one over, doesn't show it here, but it's right in this area. Both of those have large negative delta Gs, right? So if these have large negative delta Gs, they're going to pull the rest of the reaction in the specific direction we're interested in. Why is this important? It's important because this process needs to be regulated. And when you think about regulating a process, you regulate those reactions that have the biggest control on the process. And those reactions are the ones with the large negative delta Gs. So the carboxylation reaction, the reduction reactions, and these two phosphate-releasing reactions are the ones that are controlled. Why should we control this? Why should there be times when the dark reactions are shut off and times when the dark reactions are functioning? You mean?
Just the water flows, and as you explained, like oxygen will diffuse through, but not carbon dioxide.
Okay.
You don't want the particle cycle to be.
Why not? Why don't you want this cycle working if there's no CO2?
Because it'll waste ATP and NADPH.
Well, could it use ATP and NADPH if there's no CO2? Remember that first, the slide that I skipped by? That one. If there's no CO2, can we make ATP and NADPH? This is the thing we talked about at the beginning, spent too long talking about at the beginning of the last lecture. You can't, right? Because if you don't have any CO2, then you can't use up the ATP and NADPH that the light reactions have produced. You can't regenerate the ADP and NADP the light reactions need to function. Right? Without NADP, now you know the electrons and electron transport, the light reactions have got nowhere to go. Right? So we don't. What you're saying is correct. The dark reactions are shut off under certain circumstances, but there's a more important time that it's shut off. Under what circumstances would you expect the dark reactions, the Calvin cycle, to be turned on? Yeah, during photosynthesis. When would you expect it to be turned off? When it's not. Okay, when does photosynthesis not happen? At night, in the dark, right? Okay, why? Why would you want to have this turned off at night? This is a little more complicated question. Why would it make sense that you would regulate the enzymes involved in CO2 fixation to keep them shut off at night? Anna?
Are there other processes that use the NADPH and ATP?
Yeah, that's basically it. Where at nighttime does ATP and NADPH come from? We haven't talked about this yet, but I think you probably know the answer. Yeah, respiration, right? Right? And so to make that ATP and NADPH, you're using up sugars, right? Okay, so does it make sense to take sugars, make ATP and NADPH, use that ATP and NADPH to make sugars, that then you're going to break down again, what's going to happen? It's a futile cycle. You lose energy, right? So that's why the Calvin cycle is shut off in the dark, because it would create a wasteful cycle where energy in the plant would just be turned into heat. There would be no net production of anything other than heat. Right? So the Calvin cycle is on in the light and off in the dark. The question is, what are the key things that regulate this process? What are the things that turn on and turn off the enzymes associated with the Calvin cycle? And one of the main ones has to do with redox-dependent reactions. So one of the things that photosynthesis produces, the light reactions, are reduced compounds, NADPH, right? And NADPH can be used to, directly or indirectly, to drive other redox reactions. And one of the main redox reactions is that many enzymes contain disulfide bridges. There's an amino acid called cysteine. So here's a protein, and here is the SH group of a cysteine amino acid sticking out. And this can be, let's see, when these two cysteines are oxidized, they form a covalent bond between them. That's called a disulfide bridge. And when you reduce them, it goes back the other way. This is one of many mechanisms that is used to regulate enzyme activity. This one may be off and this one may be on, or vice versa. But basically, the formation and breaking of that disulfide bridge is like a metabolic switch that turns the enzyme on or off. So, which one is going to be active in photosynthesis? The oxidized or the reduced form? Don't look. I want you to use your brains. If the light is on, are we producing an excess of reductant, or are we using up the reductant? Are we producing reductant in the light? Yeah. So which way is this reaction going to be favored to go in the light? Towards this form or this form? This way, right? Towards the SH, right? So these is, in photosynthesis, this is the active form, because the light produces reductants that can drive the reaction in this way. In the dark, that reductant goes away. Molecular oxygen is sufficient to oxidize these back again. So the only thing we really need to think about is how do we get energy from NADPH to oxidize and reduce these various compounds. And it's done by sort of a little mini electron transport chain. Ferredoxin is actually an electron carrier near photosystem 1. And electrons from ferredoxin can be used to reduce a small protein called thioredoxin. Thioredoxin turns out to be an important redox-dependent regulatory molecule in all organisms, not just in chloroplasts, but in all organisms. And the thioredoxin, the electrons, the reducing potential in thioredoxin can be used to convert an enzyme from its inactive oxidized form to its active reduced form. So in the light, we're driving this reaction towards the active state. And in the dark, when we take away the reductant, oxygen converts this back to the inactive state. So that's the main mechanism by which most of the key enzymes in the Calvin cycle are regulated, by reductant that is a product, the normal product of the light reactions. That reductant reduces the disulfide bond to activate the enzyme. This is true for all the main enzymes in the main regulatory enzymes in the Calvin cycle, except for the most important one. And that most important one, we'll finish up with this. the key enzyme that's involved in the carboxylation reaction. So that takes CO2 plus RUBP and makes 2 PGA. The enzyme that catalyzes this is called Rubisco. It's short for ribulose bisphosphate carboxylase oxygenase. Don't worry about the details of it. It's in the book. You can read it. We'll talk about the different enzyme activities it has. Rubisco is a pretty interesting enzyme. It's the most plentiful protein on Earth. There's more Rubisco than any other protein on Earth. And you should ask yourself why. Why does there have to be so much Rubisco? Well, I think we mentioned already one aspect of this. Rubisco is a crummy enzyme. It's a really bad enzyme. It doesn't work very well under the conditions that it's functioning under. So, first of all, if we look at Rubisco and ask how much Rubisco is in the stroma of a chloroplast, Rubisco is about 4 millimolar. 4 millimolar protein catalyst. That's higher concentration than most of the intermediates in the Calvin cycle. There's just a ton of it there. The chloroplast, the stroma of the chloroplast is Rubisco soup. Have any of you ever isolated proteins from plants besides photosynthetic proteins? You ever done a protein purification? No one's done that? One of the biggest problems with protein purification in plants is contamination by Rubisco. There's just so much of it there. It's really hard to get rid of. Okay, so why is there so much of it there? What makes it such a bad enzyme? Okay. Couple things. First of all, we talked about enzymes bind their substrates, convert it to products, release the products, and then they're ready to go again. That's called the catalytic cycle. So going from binding products, binding reactants, producing products, going back around and starting over again, that's a catalytic cycle. Typical catalytic cycle for an enzyme is 1,000 per second. Fast enzymes would be 1,000,000 per second. Rubisco is 2.5 cycles per second. It is slow. What are the key substrates for Rubisco? CO2. If we plot, we do a Michaelis-Menten plot of rate of CO2 fixation versus CO2 concentration. It has a typical sort of thing like this, right? So remember we said that the Km was the rate, or the concentration at which the rate is 50% of maximum. The Km for Rubisco is about 20 micromolar CO2. Sorry, 20 micromolar CO2. Anybody know what the ambient concentration of if you had cytoplasm in equilibrium with the air, what's the concentration of CO2 in the cytoplasm? You probably don't know, but. Very small. Yeah, it's down here. Ambient is around 10 micromolar. What does that mean about Rubisco as far as CO2 is concerned? Yeah, it's run slowly because CO2 is limiting, right? So we've got slow catalytic cycle, very poor affinity for its key substrate, and on Thursday we'll add one more thing to this, that Rubisco has it can catalyze another reaction besides this, where it uses oxygen instead of CO2. I think I actually have yeah. So this is the desired reaction, taking ribulose bisphosphate, adding CO2 and making 2 PGAs. But it turns out Rubisco catalyzes a competing reaction, an oxygenation reaction, that takes ribulose 1,5-bisphosphate and produces 1 PGA and 1 2-carbon molecule phosphoglycolate. Okay? And to sort of lead us into thinking about why this is a problem, first of all, it's competing with the reaction that we want. Second of all, glycolate is toxic to cells. So we've got this competing reaction that is preventing some of the desirable reactions from happening and producing a byproduct that the cell has to deal with in some way. Okay, so we'll come back to talk about this, start with this on Thursday. Okay?